in Australia, Monday over there, Sunday here in Seattle. And I am a postdoc from the Fusobacterium lab. And today I'm going to talk about the bacterial-host interactions in the context of the tumor microenvironment. And this is the outline of the talk today. I'm going to mention a brief introduction about how the tumor-associated microbiota is associated with cancer progression. And then I'm going to focus in my findings during my postdoc. And I have 3 chapters of results. The first one is to evaluate where the microbes are located in the tumor tissue and what is these microniches that bacteria prefer to colonize. Second, I'm going to introduce a new method that is called the basic approach that allow us to identify whole cell populations that contain bacteria and the associated transcriptome at the single cell level resolution. And third is a project that I am developing right now that is about how bacteria that are found in the extracellular space, they can interact directly to the epithelial cancer cells and that can induce cancer progression. And then I'm going to mention some conclusions and the acknowledgements. So first, I'm going to introduce Fusobacterium nucleatum. That is a gram-negative bacterium that is present in the oral cavity in the healthy individuals and is part of the normal microbiota in the oral cavity. And it has been associated with periodontal diseases. However, in 2012, a microbiome analysis based on 16x ribosome RNA sequencing has shown that in the colorectal cancer tissue is colonized by few microorganisms that belong to the Fusobacterium nucleatum. And as you can see here, the composition of these bacteria is a bit different from the normal column from the blue, where you have a highly diverse composition of the bacteria species. And because of this study, it showed that these microbes that are enriched in the colorectal tissue is directly associated with cancer progression. And then later, by using imaging techniques, the authors found that the tumoral bacteria is found intracellular and they can interact with cellular elements from the immune system and the epithelial cancer cells. And in this study, it showed that in some bacteria communities, it can induce DNA damage as a molecular mechanism to promote cancer progression. And this bacteria, this is called Escherichia coli, that produce this toxin, that is the PKS toxin. They can directly induce DNA damage, and you can see it here by the staining against phosphorylated gamma H2X. That is a histone that gets phosphorylated when you have DNA damage. And then in these clinical studies, show that in patients that have higher loads of Fusobacterium nucleatum is correlated with poor clinical outcomes and is resistant to chemotherapies in comparison to patients that have lower loads of Fusobacterium nucleatum that have better prognosis and they can be sensitive to the chemotherapies. And this paper in Science, my mentor, Susan Bullman, has shown that in colorectal cancer, Fusobacterium nucleatum can be also detected in the corresponding liver metastases, suggesting that Fusobacterium nucleatum can be associated with metastasis and cancer colonization to secondary organs. And this is the model that we are working actually. This is the tumor microenvironment. That is a very complex tissue that is not only composed by malignant cells, but they also contain other cell populations like fraulas, endothelial cells in the vasculature, and you can also have different immune infiltrates and different immune cell populations. And on top of that, the tumor-associated microbiota is also a fundamental component of that tumor microenvironment. They can directly influence the cell functionality from other elements of the tumor tissue. And in postdoc, I was aiming to evaluate where these microbes are located in the tumor tissue and what is the microniches that bacteria prefer to colonize, and also what is the transcriptomic and the cellular alterations that are associated with intratumoral bacteria. So then I'm going to show you some of my findings on my postdoc, and is to see where these microbes are located in the tumor tissue and what are these microniches that bacteria likes to colonize. So first, in order to answer these questions, we implement this novel technique that is called the 10x Visium spatial transcriptomics. And in this approach, we have identified colorectal cancers that contain bacteria by using NRSCOP. And then we allocate these pieces of the tumor that contain bacteria in the next visual slide that have 4 capture areas. And in every capture area, there is an array of dots, and in every dot has an array of probes that can capture the RNA when the tissue is permeabilized. And it's worth to mention that every probe has a unique molecular identifier, meaning that you can get the original transcriptional load of the transcripts after the amplification steps, and you have the spatial barcode that gives you the spatial coordinates for every transcript. Then, after different amplification steps, the transcripts are aligned to human databases in order to generate the human libraries, and the reads that cannot be aligned to these databases are going to be aligned to the microbial databases in order to taxonomically identify the bacterial communities that are contained in these tumors. And this is the data that we get. In the left side, you have the RNA-scope that show the distribution of bacteria in the tumor tissue. And because the probes can also acquire or capture 16x ribosomal RNA bacterial transcripts, you can actually taxonomically resolve all the bacterial communities that are in these tissues in an unbiased and untargeted way. And in this technique, you can also map the distribution of the bacterial transcripts. And you can see here in this example, all the bacterial transcripts are located in a specific microniche in the tumor tissue. And this localization of these transcripts coincide with the RNA-scope image that you can see in the left side of the presentation. And by using this technique, you can also map the distribution of particular bacteria species like Fusobacterium and Parvimonas in the head and neck case. And in the colorectal cancer case in the bottom, you can observe the distribution of Fusobacterium and Bacteroides. And this is a colorectal cancer case, and this is an RNA-scope image. And you can see here that bacteria seems to colonize a specific microniches in the tumor tissue. And you can see here the distribution of Fusobacterium nucleatum in red. And we can also target the 16S ribosomal RNA in order to visualize the distribution of all the bacteria communities in the tumor tissue. And you can see here, in this particular case, there are like large areas of the tissue that are colonized by bacteria. As you can see here in red and cyan, in comparison to other areas of the tumors that contain lower levels of bacteria or doesn't contain bacteria at all. And what that tells us is that bacteria in the tumor are highly organized in specific macroniches in these tumors. Next, we want to characterize the macroniches that are colonized with bacteria, or why bacteria colonize preferentially these macroniches. And in order to answer these questions, we implement another technique that is called the digital spatial profiling that was developed by NanoString here in Seattle. And by using this technique, we have isolated tumors from colorectal cancer patients, and then we embed it in formalin blocks, and then we generate 2 sequential slides. The first one, we did RNA-scope in order to visualize the distribution of bacteria across the tumor tissue, and the second slide, we did immunohistochemistry in order to identify the cancer compartment that is positive for pancreatic creatine and the immune segment that is positive for CD45. And in this slide, we incubate with a panel of 77 antibody panels that target molecules that are involved in the immune response and cancer development. And in this technique, every antibody is attached to a UV linker and a barcode oligo. Then when you illuminate with UV light into the areas that contain or no bacteria, the UV linker is degraded and the barcode oligo is detached, and then you can collect it and sequence, and that gives you the spatial coordinates for every protein target. And this is what the data looks like. So in the left, you have the RNA-scope image, and where we identify regions of the tumor tissue that contain or no bacteria. And in the right, you have the immunohistochemistry, where you stain against pancreatic creatine, where you detect the epithelial cancer cells in green, and the CD45 that stain the immune cell populations in red. And by using segmented analysis, you can capture the barcode oligos from the immune and the epithelial segment separately. What we found is that in the immune segment, or in the CD45 positive cells, there is an enrichment of neutrophils that are CD63 positive, and an increased upregulation of immunosuppressive molecules that are known to inhibit the T-cell response, like for example, CTL4 and arginase-1. And this technique, you can actually measure phosphoproteins like ERK and Janus kinases, indicating the signaling pathways that are activated in response to bacteria in these microniches. We can also saw that is a downregulation of T cell markers like CD44, CD40, and K67 that are meaning that these T cells that are in these microniches are less active or they are immunosuppressed and they have lower proliferation potential than other T cells from other tumor regions. In the epithelial compartment, we have a similar profile where in the bacteria, in the regions that contain bacteria, there is an increased infiltration of neutrophils that are CD63B positive and a regulation of immunosuppressing molecules like PD-1 antigenes 1, and also an increased levels of proteins like ERK1/2 and Janus kinases, also indicating the signaling pathways that can be correlated with the presence of bacteria in the microniches. And something very important to mention is that these epithelial cancer cells that populate these microniches contain lower levels of K67 and P53, meaning that these epithelial cells can display more aggressive phenotypes than other epithelial cancer cells from other microniches. And now I'm going to jump into a second chapter of the outcomes. That is like we develop a new method that is called the basic protocol that allows us to identify host cell populations that contain bacteria at the single cell level resolution. In order to develop this technique, we have isolated tumors from head and neck cancers, and then we dissociate these tumors in order to process for single cell RNA-seq techniques. And then we apply the 10x genomics chromium protocol. We modified it by adding a primer that can bind to the 16S ribosomal RNA. And by doing this, every bacterial transcript, by using the TSO, every bacterial transcript is going to contain the unique molecular identifier and the 10x barcode, assigning every bacterial transcript to the corresponding host cell in the genes. Then we just run the standard protocol from 10x genomics chromium in order to elaborate the human libraries. And then we took an aliquot from the amplified host CNA, and then by applying by introducing primers that can amplify the bacterial 16S ribosomal RNA, we could resolve the bacterial libraries. And this is just to indicate how specific is this technique to detect cancer cells that contain bacteria or that are adherent and invasive. And then we did different co-cultures with the cell line, that is the ST29 cell line, with different bacteria species that were isolated from colorectal cancer tumors. And what we found is that by using this technique, the basic approach could identify cancer cells that contain only adherent and invasive bacteria, like for example, Bacteroides, Fusobacterium, and Prevotella. And this technique could detect transcripts from Escherichia coli because this bacterium is not adhesive and is not adherent. So as a result, this technique can only identify cancer cells that contain bacteria that is adherent or is adhesive to the cancer cells. And this is just to mention that the bacterial transcripts is a minor component in the human libraries. You can see that there is below 0.02%, but when you apply the invasive approach, you have an enrichment of bacterial transcripts in the bacteria libraries almost 100 times. And this is when we apply the invasive approach to the tumors from the head and neck cancers. And you can see here in the UMA plots, you can only resolve the cell populations that are contained in these tumor tissues, but you can also identify the host cell populations that contain bacteria. In this case, the macrophages in green and the epithelial in red clusters contain most of the bacterial transcripts in these samples. And you can also evaluate the distribution of Fusobacterium that is a particular bacterium in these samples, and you can use the unique molecular identifier in order to select a population of host cells that contain a certain number of bacterial transcripts. In this example, we apply a threshold of 4 UMI, meaning that you can select a population that contains 4 or more bacterial transcripts, and this is important because you can filter out a host cell population that contain lower levels of bacterial transcript that could correspond to contaminants or artifacts from the amplification steps. And because we have introduced a primer that targets the 16S ribosomal RNA, we could taxonomically identify all the bacterial communities that are in these tumor tissues at the single cell level resolution. Additionally, you can do other type of analysis. You can do differential expression analysis comparing cancer cells that contain or no bacteria. And you can do this analysis in specific cell clusters. For example, in the epithelial cluster or in red, you can see that the cells that are associated with bacteria there is a upregulation of genes that are associated with metastasis and cancer progression. And in the macrophage cluster, the myelin cells that contain bacteria, there is an increased expression of inflammatory molecules like chemokines and other inflammatory molecules that belong to the interferon and the TNF-alpha signaling pathways. And you can also do other type of analysis, for example, signaling pathways analysis, where you can identify the signaling pathways that is associated with the presence of bacteria in this type of cancers. And then I'm going to jump into my last chapter of results. That is that we found that bacteria can be also found in the extracellular space, and that can directly influence cancer behavior in epithelial cells. So this is an overlap image with between immunohistochemistry testing against F-actin, pancyclidine, and K67. K67, as I mentioned before, is a marker for proliferation, and it's an overlap with an RNA-scope image that is stained against bacteria, the 16S ribosomal RNA, and Fusobacterium nucleatum, and when we identify regions of interest that contain bacteria or not. And you can see here that in the microniches that contain bacteria, the cancer cells have lower expression of Ki67 in comparison to cancer cells that are in the microniches that doesn't contain bacteria. And then by using higher resolution confocal imaging, we could identify that the cancer cells that are in these microniches looks very different from the epithelial cells that reside in microniches that doesn't contain bacteria. And you can see here that the epithelial cancer cells doesn't have the cell-cell interactions that are characteristic of epithelial cells. When we did neighbor analysis, we found that the epithelial cells in these microniches that contain bacteria are in a scattered conformation, meaning that the distance between every cancer cell to the neighbor cells are higher than the epithelial cells that reside in these microniches that doesn't contain bacteria. And therefore, these epithelial cells are in a more clustered distribution, meaning that the distance between neighboring cells are much lower than the epithelial cells that are found in these microniches. And I have a different more results, but in summary, we found that extracellular bacteria can digest the cell-cell interaction between epithelial cells, and that change the phenotype to a more mesenchymal and more invasive behavior. However, we use a spatial single-cell transcriptome analysis in order to identify the transcriptome of this cell population that populate the microniches that contain bacteria. And you can see here, we apply a panel of 1,000 genes, and we modify this panel by introducing probes that can target the 16S ribosomal RNA and Fusobacterium genes in order to map the distribution of bacteria in these samples. And what we found is that in the microniches that contain bacteria, there is an increased expression of inflammatory molecules like CXCL8, interleukin-1, and increased infiltration of myeloid cells that are CD11 positive. And also we can do a macroniche niche analysis that can compartmentalize or separate different niches based on the cell population of a particular niche. And we identify 4 different niches. The niche type 2 are the niche that reside bacteria. And you can see here the epithelial cells are in this scattered distribution where they don't have the cell-to-cell interactions. And you can see here in this niche type 2, you can see that the epithelial cells have lower number of transcripts in general, meaning that these epithelial cells have lower transcriptomic activity than other epithelial cells from other niches like niche type 4 or niche type 3. At the gene expression level, when we compare epithelial cells from niche type 2 to niche type 3, that are epithelial cells that are far away from the infected areas, we can see that is a downregulation of almost every gene that was contained in the panel of genes. And there is an increased expression of genes that are involved in cancer cell transdifferentiation, like the EMT, like for example, SAT1, TNF-alpha, and metalloproteinases like MMP7. And very interestingly, when we compare epithelial cells from the niche 4, that are epithelial cells that are very close to the infected areas, versus epithelial cells that are in the niche 3, that is epithelial cells that are far away, we found that these cancer cells are in the process to get differentiated to a more mesenchymal behavior with a regulation of catenin B1 and other molecules that are involved in metastasis, meaning that these cancer cells that are next to the macroniche that contain bacteria are in the process of transdifferentiation to other cell populations that are more invasive. And that's it. So I'm going to draw some conclusions. The first one is that bacteria is not randomly distributed in tumor tissue, but instead they are concentrated in specific macroniches in the tumor region. These macroniches are necrotic, hypoxic, and very immunosuppressive, with an increased infiltration of myeloid cells and exclusion of T cells. we developed a new method that's called the in-basic approach that allow us to identify host cell populations that contain bacteria and the associated transcriptome at the single cell level. And in my current and more recent project, we found that bacteria can be found extracellular, and they can directly interact with epithelial cancer cells in order to promote cancer cell invasion and cancer progression. And I'm going to thank my mentor, Susan, and all the team, especially Clara, Andy, Henry, and other past members like Kathleen and Alex. And I also work with the shared resources from the Fred Hodge, like cellular imaging, experimental histopathology, genomics, comparative medicine, and all the funded partners to make this